Hi there, welcome back. Today we're going to be covering the 3 steps of T cell activation using Th1 cells as an example. The Th1 response is powerful because it responds to viruses, bacteria, and protozoa. And because infections usually occur at epithelial barriers, dendritic cells will play a really important role in the Th1 response. Dendritic cells like to camp out like sentinels at epithelial barriers where they have the highest likelihood of encountering a pathogen. After encountering a pathogen, they then migrate to the spleen or the nearest lymph node where they present an antigen from the pathogen to a Th1 cell, and this initiates the specific immune response. The first signal is when the T cells interact with the antigen presented by the dendritic cell, and they do this through their T cell receptors. Only a dendritic cell can do this cross-presentation of antigens using both MHC1 and MHC2, and this helps them to co-stimulate both the CD4 and CD8 T cell that increases the power of their activation and their proliferation. It might be helpful to note that there are additional molecules on these T cells that bind to the MHC molecules for stabilization, but they're not pictured here just for the sake of brevity. So the second signal involves co-stimulation of the T cells to increase the level of activation by the dendritic cell. The binding of CD4 T cells actually stimulates the dendritic cell to begin producing more of these MHC1 molecules and these B7 ligands. This B7 CD28 binding is important for CD8 T cells to ensure that they don't become autoreactive and start accidentally attacking the body's own tissues. Without the second signal, the immune response cannot proceed and results in anergy. For the third signal, the dendritic cell begins secreting these cytokines called IL-12. And that's in response to either being stimulated by the CD4 T cell or other inflammatory cytokines and signals in the environment. So this IL-12 combined with the inflammatory environment helps to drive the CD4 T cell to become a fully developed T helper 1 cell with effector functions. Similarly, IL-12 can help the CD8 T cell mature into either a cytotoxic T cell or a memory T cell. IFN gamma is another cytokine that helps to activate the dendritic cell to produce more IL-12, and both help to aid in the induction of the Th1 response. Now these Th1 cells are capable of producing cytokines of their own, including IL-2, which helps to drive the expansion of cytotoxic T cells and ensures the differentiation of these memory CD8 T cells. Lastly, the dendritic cell releases IL-15, and this aids in the activation of T cells, the homeostasis of memory CD8 T cells, and proliferation of natural killer cells. It's also really interesting to know that these CD8 T cells can actually become activated on their own. They can become independently activated when there are sufficient levels of IL-2 just floating around in the microenvironment. However, if there isn't enough IL-2 in the microenvironment, then it can really compromise this independent activation and also compromise proliferation of the cytotoxic T cells and the production of the memory CD8 T cells. After activation, Th1 cells begin releasing more cytokines of their own, including IL-2 and IFN gamma. This helps to activate cytotoxic T cells to release cytotoxic granules, including granzyme and perforin, to induce the apoptosis of infected epithelial cells, or tumor cells. Both CTLs and Th1 cells can release TNF-alpha and IFN gamma, and this also helps induce apoptosis and recruit macrophages for cleanup. The cell-mediated Th1 response isn't just limited to intracellular infection because it also aids in the clearance of tumor cells. And to review, here are a few of the immune cells that are activated by Th1 cells, and they include macrophages, neutrophils, cytotoxic T cells, and B cells. You might recognize this slide from the last episode that gave a broad overview of the immune system, but it's helpful here to review the effector functions of the Th1 response, chronic inflammation, tumor immunity, and killing of intracellular pathogens. But do recall that the 3 signals required for activation of a T cell applies to every single type of T helper cell, and each one of these T helper cells, after they're activated, achieve these effector functions, and they achieve these effector functions by the release of their own cytokines that drive the specific immune response. So in summary, here are the 3 signals involved for a T cell activation. Signal 1 involves the recognition of the antigen. Signal 2 involves binding to stimulate the activation process. And signal 3 involves instructive cytokines that can complete the activation and proliferation of T cells. Well, that's it for the 3 steps of T cell activation and a quick overview of the Th1 response. If you're enjoying these videos and would like to follow along, feel free to subscribe to my channel here or go find me on patreon.com, where there'll be a little bit more exclusive content and areas of opportunity for engagement. So anyway, I appreciate you following along in this learning experience, and I'll see you on the next one.